All right, today we're going to dive deep, and I mean really deep inside our own bodies. We're going to meet the tiny secret messengers that are, and this is no exaggeration, running pretty much everything. They're called GPCRs. And these microscopic machines are, at a fundamental level, what make you, you. So, have you ever really thought about it? How does a single one of your cells, just one, know what to do from one moment to the next? How does it know to process light so you can see, or to register the scent of a rose, or to kick into high gear when you get that jolt of adrenaline? Well, the answer is this absolutely massive family of proteins that work kind of like the master switches for our entire body. They're dotted all over the surface of our cells, just waiting for a signal. And when they get one, bam, they flip the switch and tell the cell exactly what it needs to do. So let's get to know these little guys. We're about to explore these incredible master switches, which are officially known as G-protein coupled receptors. Yeah, it's a mouthful, so we'll just call them GPCRs. Okay, so what is a GPCR? The best way to think about it is like a cellular doorbell. It's a protein that's stuck right in the wall of a cell. Its job is to detect all the messages happening outside the cell, like a hormone or a neurotransmitter floating by. And it rings that doorbell to let the inside of the cell know that something important is going on. And their to-do list? It's just staggering. I mean, we're talking about the absolute fundamentals of being alive. Everything from sensing light, smells, and tastes, to responding to hormones, to regulating your heartbeat, controlling your immune system, and even managing how your body uses energy. They are completely essential. So, how does this tiny little doorbell actually pull it off? Well, its design is just brilliant, which is how it got the nickname the seven-loop signal catcher. See, this protein doesn't just sit on the cell's surface. It actually weaves its way back and forth through the cell's outer wall, the membrane, 7 times. It's this super clever design that creates a perfect little pocket on the outside to catch a signal and a corresponding part on the inside that's ready to shout that message into the cell. It's an incredibly slick piece of engineering, and the whole thing works like a beautifully simple chain reaction. First, a signal arrives from outside the cell and docks onto the receptor. That's step 1. For step 2, that docking causes the receptor to literally change its shape. It activates. Step 3, that shape change unleashes a special messenger inside the cell called a G-protein. And finally, for step 4, that G-protein just takes off like a shot to carry out a command, triggering the cell's response. Simple as that. Okay, but here's where it gets even cooler. That G-protein messenger doesn't just send one generic, boring message. Nope. The cell has a whole menu of different commands it can send out. And this is that menu of commands. We can basically group these G proteins into 4 main families. You've got the GS family, whose main job is to yell, speed up. Think of an adrenaline rush. Then there's the GI/O family, which does the exact opposite. It says, slow down, which is how things like opioid painkillers provide relief. The GQ/11 family shouts, mobilize, releasing calcium for things like muscle contraction. And finally, the G12/13 family is all about rebuild, controlling the cell's actual structure. But biology is almost never that simple, right? A lot of these receptors are, and this is the actual scientific term, promiscuous. This just means that a single receptor isn't loyal to just one type of G protein. It can activate several different pathways at once, creating this unique signaling fingerprint that allows for an incredibly fine-tuned response from the cell. So with all these speed up and mobilize signals firing off all the time, you have to wonder, what stops it? How does the cell know when to quit and not just run itself into the ground? Well, that's where the cell's braking system comes into play. And this braking system is another group of proteins called beta-arrestins. Their whole job is to step in after a signal has done its thing, shut down that receptor, and stop it from going haywire and overstimulating the cell. They are absolutely essential for keeping everything in balance. And this is no simple brake pedal. It's a really sophisticated system. It can desensitize the receptor, making it less sensitive for a bit. It can also physically pull the receptor right off the cell's surface, a process called internalization, to basically put it in timeout. And get this,
Beta-arrestins can even send out their own separate signals, adding a whole other layer of control.
So, all this intricate microscopic machinery, why should we care? Well, because understanding it isn't just a fun science fact. It is completely revolutionizing modern medicine as we speak.
Okay, get ready for this number. Right now, about 35%, that's more than a third, of all FDA-approved drugs on the market work by targeting these exact GPCRs.
This chart really puts that into perspective. That first bar, the big one, that represents all the drugs that target this one single family of proteins. That second bar, that's all the drugs that target every other type of protein in the human body combined. The fact that one family dominates so much of pharmacology, it just shows you they are the most important drug targets we have, period.
But here's the real kicker. Even with all that success, as this quote points out, we have barely even scratched the surface. There are hundreds of other GPCRs in our bodies that we haven't even touched yet. The therapeutic potential that's just sitting there waiting to be unlocked is just immense.
And all this new knowledge is leading to a much smarter way of designing drugs. You can think of it like this. Old drugs were basically master keys. Sure, they unlocked the receptor, but they also unlocked all the other pathways, which often led to nasty side effects. The new goal is to create biased drugs. Think of them as precision keys, engineered to unlock only the helpful pathway we want while leaving the others locked. It's just smarter medicine.
So all this incredible research into our body's secret messengers has brought us to a really exciting new frontier. We're learning how to trade in our cellular master keys for precision tools, which leaves us with this incredible, inspiring question. As we get better and better at designing these smarter keys, which diseases will we finally be able to conquer next? Thanks for joining me.